In today's video, we'll be studying sperm transport and fertilization. And here we have the outline of all we'll be discussing. The sperm transport through the upper vaginal area, through the cervix, the uterus, the fallopian tube. We'll be discussing capacitation, fertilization, under which we'll be considering the acrosomal reaction, the prevention of polyspermy, and the fusion of the pronuclei. Now let's start off with the sperm transport through the upper vaginal area. During copulation, semen is deposited into the upper vagina of the woman. Okay, and this semen or ejaculate is made up of about 10% sperm or sperm cells, is made up of 75% fluid from the seminal vesicles, and about 20% fluid from the prostate gland, also known as the prostatic fluid. Now, the fluid from the seminal vesicle is majorly made of fructose. Okay, it contains fructose, which is the principal energy source for the sperm cells. It also contains prostaglandins. The fluid from the prostate gland contains acid phosphatase. It contains citric acid as well as zinc and magnesium ion. Now, as I stated earlier, during copulation, the semen is deposited into the upper vagina of the woman, and once it's deposited into the upper vagina of the woman, it coagulates within 1 minute of its deposition into the upper vagina. It coagulates, okay, so it forms a coagulum. This formation of the coagulum is in order to protect the sperm cells. It's important for us to take note of the fact that the semen is slightly alkaline in nature, having a pH of about 7.2 to 7.8. Okay, this is, so to say, the optimal pH for sperm activity, and the upper vaginal area has a pH which is highly acidic. So in order for the sperm to be able to survive in this highly acidic environment of the upper vaginal area, it forms this coagulum. And the formation of this coagulum, which is a loose gel, so the coagulum is a loose gel. The formation of this coagulum is facilitated by the presence of semenogelin. Semenogelin is a protein that is contributed by the seminal vesicles. Now, in this playlist, we have a video on the female reproductive system and the male reproductive system. The prostate gland and the seminal vesicle, as well as the bulbourethral gland, are all accessory organ or accessory glands of the male reproductive tract. Okay, so it's important for us to take note of that. Now, as I stated, it forms this coagulum or this loose gel that helps to protect the sperm cells from the acidic environment of the upper vagina. In addition to this coping mechanism, so to say, of the sperm cell, the fluids or the secretions in the semen also have a buffering capacity. Now, this buffering capacity of the semen is able to change the pH of the upper vagina from about 4.3 to about 7.2. And this buffering capacity lasts for about 1 minute in humans. Okay, but this 1 minute is enough time for the sperm to escape from the vaginal microenvironment to the cervix. It's important for us to take note of the fact that the acidity of the upper vaginal area is bactericidal in nature, so it helps to prevent bacterial infections from getting to the upper reaches of the female reproductive tract. This, in summary, is the sperm transport through the upper vagina. Okay, so it's important for us to take note of that. Now let's take a look at sperm transport through the cervix. But before we consider that, it's important for us to take note of the fact that once the semen is able to cross the vaginal area to the cervical canal or to the cervical area, an enzyme known as the prostate specific antigen is able to degrade this loose coagulum, this loose gel like coagulum that was formed in the vaginal area. Okay, it's called the prostate specific antigen. And this, of course, is contributed by the prostate gland. So it's important for us to take note of that. Now, let's take a look at how the sperm is transported through the cervix. Okay, now in the cervix, we have the cervical mucus. Now, the cervical mucus is the major barrier to sperm transport in the cervical canal or in the cervix. It's important for us to take note of the fact that from day 9 to day 16 of the menstrual cycle, the cervical mucus has a higher water content. Now, the cervical mucus is made up of mucin and various polysaccharides, and it's readily impenetrable. Okay, however, within day 9 to day 16 of the menstrual cycle, this cervical mucus has an increased water content, making it less viscous and more penetrable to the sperm cells. Okay, now the mucus at this point in time during the menstrual cycle is usually referred to as the E mucus. Now, after ovulation, okay, which is after this period of 9 to 16 day of the menstrual cycle, after ovulation there is a decreased water content in the cervical mucus, and so it becomes more viscous, making it almost totally impenetrable to sperm cells. It's important for us to take note of the fact that this actually serves as a form of natural family planning, the impenetrable nature of the cervical mucus after ovulation, because it's almost totally impenetrable to the sperm cell, making the chance of pregnancy very low at this period of time. Another very important fact we need to take note of is that the secondary oocyte, which would generally call the ovum or the egg, is only viable for about 24 hours after ovulation. 12 to 24 hours after ovulation. Now, after this period of time has elapsed, and if fertilization does not take place, the oocyte, the secondary oocyte or the ovum becomes degenerated. Now, going back to the sperm transport through the cervix, we have seen that the major barrier to the sperm transport in the cervix is the presence of the cervical mucus. It's also important for us to take note of that there are various immunological strategies put in place to protect the female reproductive tract from infections. These immunological strategies could be cellular, where we have neutrophils and macrophages, and we also have certain immunoglobulins like immunoglobulin A and immunoglobulin G. In as much as these immunological strategies exist, the sperm cells do not stimulate this defense mechanism. So it's very unlikely that the sperm would trigger an immunological response and then hinder its further transport into the female reproductive tract. However, after a period of about 24 hours, the sperm cells could actually be phagocytized at the region of the cervix. Okay, so let's go back to our outline and see what we have. So we have considered sperm transport through the upper vaginal area, through the cervix. Let's take a look at sperm transport through the uterus. Now, earlier on, when we were considering the semen and its constituents, we did consider that fluid from the seminal vesicle contains fructose as well as prostaglandins. Now, these prostaglandins are able to stimulate muscle contraction, and because the uterus, among other things, is made of smooth muscle in the myometrium, the prostaglandin is able to initiate a contractile activity in the uterus, and this helps to facilitate sperm transport through the uterus to the fallopian tube. Okay, so it's important for us to take note of that. In our video on the female reproductive tract, we considered the fallopian or uterine tube and the three major divisions. Okay, so the part of the uterine tube that is closest to the uterus is the isthmus. Then we have, of course, the ampulla, and then we have the infundibulum and the fimbria or the fimbriated part of the uterine tube, which is closest to the ovaries. Now, from the uterus, the sperm cells move towards the fallopian tube. Here in this picture, we can see a cross section of the female reproductive tract. Here we have the upper vaginal area, and then we can see the region of the cervix, then the uterus, and then towards the fallopian tubes. Now, it's important for us to take note of the fact that during the ovarian cycle, we have the dominant ovary. Now, in a woman, there are two ovaries. Okay, so a woman has a pair of ovary, one on each side of the body. It's a bilateral structure. Now, during each ovarian cycle, there is the dominant ovary. The dominant ovary is the ovary in that ovarian cycle that produces the dominant follicle. Essentially, it's the ovary that releases the ovum in that ovarian cycle, and it alternates. Okay, so for the first ovarian cycle, just as an instance, we could have this ovary releasing the ovum, and then for the second ovarian cycle, we could have this ovary releasing the ovum. So we have the dominant ovary, the dominant follicle, and by extension, the dominant uterine tube or the dominant fallopian tube. Now, the dominant fallopian tube is where the ovum would be after it has been released. So, after ovulation, the dominant fallopian tube or the dominant fallopian tube that is ipsilateral to the dominant ovary. Ipsilateral is simply a fancy term for on the same side. So the fallopian tube that is on the same side as the dominant ovary would have the oocyte, would have the secondary oocyte within it, usually moving towards the uterus, but around the ampulla region of the uterine tube. Okay, so it's important for us to take note of that. Now, after the sperm has traveled from the upper vaginal area through the cervical canal and through the uterus, how exactly is the sperm able to tell in which direction it's to go to? Okay, so it's almost like the sperm transverses through the vaginal area, transverses through the cervical canal, transverses through the uterus, and then it gets to this T junction, and it's like, which way do I go now? Okay, do I go to the A direction or the B direction? Now, it's important for us to take note of the fact that the sperm cell actually has certain olfactory receptors. Okay, these are olfactory receptors, and they are also sometimes called odorant receptors. Now, these odorant receptors of the sperm cell are able to detect or, in quotes, perceive progesterone. So they are able to detect cumulus derived progesterone. That is the progesterone that is derived from the cumulus oophorus of the released oocyte. These olfactory receptors of the sperm cell are able to detect these chemoodorants as well as other chemoodorants. It's also important for us to take note of the fact that certain studies have shown that the dominant ovary or the dominant fallopian tube has a temperature difference of about 1.6 degrees Celsius from the fallopian tube that is contralateral. I'm using some anatomical terms here. Contralateral means on opposite side. Okay, so basically, if we have it that the ovary on this side, illustrated in this picture, B. So the ovary on the side B is the ovary that has released the ovum for this particular ovarian cycle. Studies have shown that the fallopian tube that is ipsilateral or on the same side as this ovary has a temperature difference of about 1.6 degrees Celsius higher than the fallopian tube on the other side, which is the fallopian tube that is contralateral. Okay, fancy term for on the opposite side. So the sperm cells are also able to detect this thermal stimulus, and so there is a preferential pathway. There is a preferential pathway or passageway of the sperm cell to the dominant fallopian tube. Okay, so there are two major factors that determine or helps the sperm cell, so to say, to make the right decision on where to go once it gets to this T junction. So once it gets to this T junction, it's like, do I go left or do I go right? Two major decisions or two major factors that influence that decision. We have the chemoodorant, where we said that the sperm cells have these odorant receptors and they are able to perceive, so to say, progesterone and other chemoodorants, and they also make the decision or the thermal decision where they are able to sense that difference in temperature, that thermal stimulus, and so they have a preferential movement towards the dominant fallopian tube. Of course, this is not to say that some sperm cells do not move in the opposite direction, okay? That's not to say that they don't move in the opposite direction, but this is under normal condition, ceteris paribus. This is what happens when the sperm cell is traveling towards the uterine tube. Now let's go back to this labeled image of the uterine tube. In this labeled image of the uterine tube, we can see that the part closest to the uterus is the isthmus. And it's important for us to take note of the fact that the sperm cells actually accumulate at this region. So they have a gathering, basically. They come together and they gather at this isthmic region. They essentially bind to the epithelium of the isthmus. Okay? So they bind to the epithelium of the isthmus. And when they bind to the epithelium of the isthmus, the isthmic secretions of the tubal epithelium of this region they stimulate capacitation, basically, in the sperm cells. Now, capacitation is a very important process that must take place in sperm cells in order for them to be able to fertilize an ovum. Essentially, put very simply, freshly ejaculated sperm cells are actually unable to fertilize an oocyte. So even if the sperm cell is directly is released, it's still unable to fertilize an oocyte until it undergoes capacitation. Okay, now let's take a look at capacitation. So far we have considered the sperm transport through the upper vaginal area, through the cervix, through the uterus, and also through the fallopian tube. Now let's take a look at capacitation. What exactly goes on during this process? Now, there are two major events that take place during capacitation. The first is the removal of cholesterol, and the second is the removal of some membrane proteins and glycoproteins. Now, in our video on the cell plasma membrane or the cell membrane, we discussed the cell membrane, explaining that the cell membrane is made of this phospholipid bilayer. Okay, so we had something like this, we had the head and tail orientation, something like this, and we mentioned a very important theory, so to say, and it was the fluid mosaic theory of the cell membrane. Basically, this theory explained that the cell membrane is somewhat fluid in nature. Okay, it's somewhat fluid in nature such that the membrane proteins can actually laterally drift and diffuse through the plasma membrane or through the cell membrane. Okay, so we mentioned that idea, and alongside this idea we also mentioned of how cholesterol serves as a fluidity regulator, where we said under certain conditions, cholesterol could potentially stiffen the cell membrane, and under other circumstances, it could cause for more fluidity within the cell membrane. Now, it's important for us to take note of the role of cholesterol in the acrosomal membrane. The acrosomal membrane is basically the membrane that surrounds the acrosome. Okay, the acrosome, as we can see in this picture that labels the sperm cell, is this region here. Okay, and it contains certain proteolytic enzymes. These enzymes, as we would go on to see, would be what would help the sperm cell to penetrate the zona pellucida. The enzymes contained within the acrosome are highly digestive, highly proteolytic. So the cholesterol within the acrosomal membrane basically helps to somewhat stiffen that acrosomal membrane, such that it prevents any form of leakage of the acrosome or the acrosomal enzymes. Okay, it prevents any form of leakage that could occur from the acrosome to the exterior of the sperm cell. So it gives the acrosomal membrane a toughening, preventing any form of movement of the acrosomal enzymes. Now, for the membrane proteins and the glycoproteins, these membrane proteins are glycoproteins, some of which were attached in the epididymis of the man, would be removed during capacitation. So capacitation, as we can see, basically capacitates, or it gives the sperm cell the capacity to be able to fertilize an ovum. Okay, and it does this by the removal of cholesterol and the removal of some membrane proteins and glycoproteins. Basically, it helps the sperm to be more mobile. It increases its mobility as well, because of course the sperm cell is able to so to say shed off some weight. Okay, shedding off the weight of cholesterol, shedding off the weight of some plasma proteins, and so it's more flexible, more mobile, able to move. And of course, we stated that this process of capacitation is stimulated by secretions from the tubal epithelium of the isthmus. Now, why is all of this so important? Why is it so important to have this cholesterol that potentially prevents leakage of acrosomal enzymes? As I stated earlier, these acrosomal enzymes are highly proteolytic, and in a situation where they are not properly confined to the acrosome, they could leak away from the sperm and potentially digest the male reproductive system. So this is very important. Once it gets to the female reproductive tract, then capacitation takes place, giving the sperm cell the capacity to fertilize the oocyte. Now, following capacitation is a period of hyperactivity. This period of hyperactivity is characterized by increased mobility of the sperm cell. So the sperm cell, just after they are capacitated, or they go through this period of hyperactivity where they are very mobile, very active, very hyperactive, and so they are able to break off from the isthmic epithelium. Remember, we stated earlier that first of all, they come together, they gather, they accumulate at the isthmus, they form a bond with the isthmic epithelium. Now, during this period of hyperactivity, those bonds are broken, and so the sperm cells are free to move from the isthmus to the ampullary region where fertilization mostly takes place. Now, it's important for us to take note of the fact that per time, only a small amount of sperm cells are actually released from the isthmus to the ampulla. Okay, so basically we have the sperm cells, they are all accumulated here, and then there's this period of hyperactivity and they break the bond. This all of this diagram is actually in the interior. This is what is going on within, not outside, but within the isthmus. And so they undergo this period of hyperactivity and they break their bond with the isthmic epithelium, and so they are able to penetrate through the isthmic mucus There's also an isthmic mucus, just as we have the cervical mucus, and the hyperactivity of the sperm cell at this time actually enhances its transport through the isthmic mucus to the ampulla or the ampullary region of the fallopian tube. Now, fertilization mostly takes place at the ampulla. So once ovulation has taken place, the the secondary oocyte or the ovum would be casually moving through the fallopian tube and probably until it gets to the sperm cell. Okay, so that's basically sperm transport in the female reproductive tract. So far we have considered sperm transport entirely through the female reproductive tract and capacitation. Now let's take a look at fertilization and the processes that take place there. Now before we do that, it's important for us to take a look at this diagram that we have here. Now in this diagram we can see the progressive reduction in the number of sperm cells up until they eventually get to the fallopian tube. Now, in a semen or in an ejaculate of about 2 to 6 milliliters, there are about 40 to 250 million sperm cells. However, the transport of the sperm from the upper vaginal area to the uterine tube or to the fallopian tube causes for a progressive reduction. As we can see, there are a lot of barriers to be crossed, a lot of things to be encountered. So basically, we can see that when the sperm cell is deposited in the upper vagina, because of the high pH of that region, initially we have the number of sperm cells to be in the order of 10 raised to the power of 7. Okay, so we have 10 million basically. It's in the order of 10 million sperm cells. Of course, this could be 5 times. Then it gets to the cervix. Then the passage through the cervix, we have the slow and the fast phase of this passage, where it decreases to the order of 10 to the power 6. Then we have the passage through the uterus, where it decreases to the power of to 10 to the power 5. Then we have the entry into the uterine tube, where it decreases to 10 to the power 4. Then we have the passage up the uterine tube by swimming and contraction of the tube. Now we can see only a small number of the sperm is actually released per time from the isthmus to the ampullary region. This is approximately in the order of 10 to the power 2. So we can see the progressive decrease as it goes on from about 10 million to nearly 100, slightly 1,000. Okay, now let's take a look at fertilization. Fertilization refers to the series of processes or the series of events that lead to the fusion of the male pronucleus with the female pronucleus. Okay, so it involves a series of events, a series of processes, rather than a single event that lead to the fusion of the male pronucleus with the female pronucleus. Now let's take a look at each of the processes involved in fertilization, one after the other. Now in our video on oogenesis, which we have in this playlist, the video on oogenesis, which basically the sequential set of events that occur through from the transformation of the primordial germ cells to the formation of the secondary oocyte. And in that video we also saw that the secondary oocyte is released at the period of ovulation, where it is arrested in the metaphase 2 of meiosis, of the second meiotic division. Now it's important for us to take note of the fact that the ovum is not formed. In other words, the second meiotic division does not progress unless fertilization takes place. So if fertilization doesn't take place, the secondary oocyte is degenerated at the metaphase 2 of the second meiotic division, it doesn't proceed to the subsequent phases, okay, unless fertilization takes place. Now here in this picture we can see the released oocyte. Okay, the oocyte is released with some follicular cells. So we have something like this, where this would be the plasma membrane of the oocyte, the secondary oocyte, or the ovum. Okay, this would be the plasma membrane of the ovum, and then we would have a paravitelline space. Then we would have the zona pellucida just around it in this way, represented in blue, and then over the zona pellucida we would have these follicular cells that form the corona radiata. Alright, so here in yellow we have the corona radiata, then just beneath it we have the zona pellucida, then we have the paravitelline space or the vitelline envelope, and then we have the plasma membrane of the ovum, and right here we can see that the secondary oocyte, which we're referring to as the ovum, is arrested at the metaphase 2 of the second meiotic division. We can see the number of barriers that the sperm cell would have to pass through in order to get into the cytoplasm of the ovum. Okay, so it would have to pass through the barrier of the corona radiata, then the zona pellucida, the paravitelline space, which is not essentially a barrier, and then finally it would have to get to the plasma membrane of the ovum. Now let's consider each of these barriers one after the other, starting off with corona radiata. Now the corona radiata is made of multiple layers of follicular cells. We have all of these follicular cells surrounding just multiple layers of these cells. And in between these cells we have an intercellular matrix. Alongside other substances, the intercellular matrix of this follicular cell is made of hyaluronic acid. Okay, now here's where the sperm cell comes to play. The head of the sperm is able to release hyaluronidase. Now hyaluronidase is an enzyme which is able to break down hyaluronic acid. Okay, and by virtue of breaking down hyaluronic acid, the sperm cell is able to weave through the corona radiata, also facilitating its passage through the corona radiata is its wavy movement. Okay, so its movement also aids its penetration through the corona radiata. Now, after passing through the corona radiata, we have the zona pellucida. The zona pellucida is a glycoprotein layer. In other words, it has this long polypeptide chain, and specifically for the zona pellucida, it has 4 major types of glycoproteins. It has the ZP1 to the ZP4. ZP2 and ZP3 of the zona pellucida form long filaments. Okay, so they form this long lattice network of filaments, and ZP4 and ZP1 form cross bridges between the filaments. So something like this would be ZP1 and ZP4. This is essentially the makeup of the corona radiata. Now ZP3 plays a very essential role because the sperm cell actually binds to the sperm receptor on ZP3. Okay, so there is a sperm receptor on ZP3, and if this be the sperm cell, the sperm binds to ZP3. Okay, and its binding to ZP3 leads to the acrosomal reaction, stimulates the acrosomal reaction. Now, what exactly takes place during the acrosomal reaction? Now, we already considered that we have the sperm cell with the head of the sperm looking something like this, and then we have this acrosome, which contains acrosomal enzymes that are highly proteolytic in nature. Okay, so we have something like this representing the head of the sperm, where the structure in red is the acrosome. Now we already established that during capacitation, we have removal of cholesterol and glycoprotein, making the sperm more flexible. But capacitation is not the same as acrosomal reaction. What happens in acrosomal reaction is this. The acrosomal reaction is actually initiated by an influx of calcium ion. So during the acrosomal reaction, it's kick-started by an influx of calcium 2 plus ions. Now this influx of calcium 2 plus ions is accompanied by an influx of sodium, okay, by an influx of sodium ion and an accompanied efflux of hydrogen ion. This efflux of hydrogen ion leads to an increase in pH. Okay, so we have an increase in pH. First, influx of calcium accompanied by influx of sodium and efflux of hydrogen ion. Now, following closely this movement of ions is the fusion of the outer acrosomal membrane. Okay, so the outer acrosomal membrane fuses with the overlying plasma membrane. Basically, we have something like this. If this be the acrosome, cap-like structure like this. Okay, now highlighted in blue would be the outer acrosomal membrane. This would be the outer acrosomal membrane, and remaining in red is the inner acrosomal membrane. Now, here in green, okay, represented here in green would be the plasma membrane of the sperm head, of the head of the sperm in green. What happens is that we have a fusion of the outer acrosomal membrane in blue with the overlying plasma membrane in green. So these two membranes fuse together. The fusion of these two membranes leads to the formation of small, tiny acrosomal vesicles. These acrosomal vesicles contain acrosomal enzymes, which are lytic enzymes, proteolytic enzymes, and when they are released, they attack the zona pellucida, and the major enzyme in the acrosomal vesicle is the acrosine. Okay, alongside other enzymes, it leads to the breakdown of the zona pellucida, and so the sperm is able to penetrate the zona pellucida and get into the paravitelline space or the vitelline envelope. Now here in this picture we can see it basically explains what I've been trying to pass across. So we can see the corona radiata, we can see the sperm cell penetrating through the corona radiata. Then it gets to the zona pellucida, and when it gets to the zona pellucida, we have the initiation of the acrosomal reaction, where the outer acrosomal membrane fuses with the overlying plasma membrane, and it releases these acrosomal enzymes that basically attack the zona pellucida and cause for the sperm to be able to penetrate or pass through the zona pellucida. Okay, now from the zona pellucida we have the perivitelline space, which is sometimes referred to as the vitelline envelope. It's a space that exists between the zona pellucida and the plasma membrane of the ovum or of the oocyte, the secondary oocyte. Okay, now after this movement from the zona pellucida through the perivitelline space, then the sperm gets to the plasma membrane of the oocyte. Now what exactly happens during this period? Now once the sperm has crossed through the corona radiata and it gets to the plasma membrane of the ovum, okay, it binds to sperm binding molecules on the ovum. These sperm binding molecules, okay, are on the plasma membrane of the ovum, and the sperm binds to these receptors basically. Now once the sperm binds to this receptor, there is a fusion Okay. Now, what happens in this case? The sperm doesn't exactly enter into the cytoplasm of the ovum. This is the cytoplasm of the ovum. It doesn't exactly enter into the cytoplasm of the ovum. Rather, the cytoplasmic content of the sperm enters into the cytoplasm of the ovum, and then the plasma membrane or the cell membrane of the sperm cell remains outside the ovum. Okay, so once it binds to this receptor, and then we have the entrance of the cytoplasmic content of the sperm. The entrance of this cytoplasmic content of the sperm leads to the formation of inositol 1,4,5 triphosphate. Now, inositol 1,4,5 triphosphate, which is mostly represented as IP3, leads to the liberation of calcium ions from internal stores. So we have calcium ions from internal stores. These calcium ions that are liberated from internal stores as a result of IP3 lead to various processes that we'll go on to see, among which we have the cortical reaction as well as the initiation or the continuation of the meiosis, of the second meiotic division. Remember, it was arrested at metaphase 2, but this liberation of calcium ion from the internal stores lead to the stimulation of the continuation of this meiotic division, as well as cortical reaction. Now, cortical reaction, as we would see, helps to prevent polyspermy, which is the entrance of more than one sperm cell into the oocyte or into the ovum. Okay, there are two mechanisms by which the ovum is able to prevent polyspermy. The first one is the oocyte membrane block. Now, as stated earlier, the sperm binds to the sperm binding receptor on the plasma membrane of the ovum. And once it binds to this receptor, there is a cascade of reaction that leads to the removal of other sperm binding receptors from the oocyte. So basically, if this be the plasma membrane of the oocyte, okay, and then we have all of these vesicles that contain or that have sperm binding receptors. We have all of these sperm binding receptors over the plasma membrane of the oocyte. Once a particular sperm binds to the plasma membrane, binds to these sperm binding receptors, we have series of reaction that leads to the removal of the other sperm binding receptors. Okay, this is to prevent polyspermy, and it's referred to as the oocyte membrane block. Now, for the corticular reaction, as I stated earlier, the entrance of the cytoplasmic content, okay, the entrance of the cytoplasmic content of the sperm cell leads to the formation of IP3, which is inositol 1,4,5 triphosphate. This inositol 1,4,5 triphosphate leads to the liberation of internal stores of calcium ions, and this release of calcium ion causes for corticular reaction. Now, what exactly occurs or what exactly happens in the corticular reactions? Now, we have these cortical granules. These cortical granules are small vesicles that contain proteolytic enzymes as well as polysaccharides. So we have them, they are just beneath the plasma membrane of the ovum. Okay, here they are highlighted in red. Now, this rapid increase in the concentration of calcium ion initiates the corticular reaction. So basically what happens is that we have a fusion of the cortical vesicles with the plasma membrane. So the cortical vesicle fuse to the plasma membrane, and their content are taken out of the plasma membrane by the process of exocytosis. So basically we have something that looks like this. This is the plasma membrane in yellow, and then we have the cortical vesicle. Due to the increased calcium liberation, it fuses with the plasma membrane of the ovum, and so we now have something that looks like this. And at the site of fusion, we have, of course, this is the cortical granule membrane, and its content is taken out of the plasma membrane by exocytosis. Now, remember, I stated earlier that what is contained in the cortical granule, we have proteolytic enzymes and then we have polysaccharides. Now, when the polysaccharides get into this perivitelline space, all of these processes occur during the cortical reaction. So this is the perivitelline space. And then we also have the zona pellucida. So this is the zona pellucida in blue. When these polysaccharides and proteolytic enzyme get into the perivitelline space, the polysaccharides are hydrolyzed. Okay, and when they are hydrolyzed, they basically increase, so to say, they increase the volume of the perivitelline space and by extension push away. So basically, we have an expansion of the perivitelline space, which pushes away the zona pellucida, taking the zona pellucida further away from the ovum. Okay, it takes the zona pellucida further away from the ovum. Some of the secretions from these cortical granules also seep into the zona pellucida. Okay, because the zona pellucida is a porous glycoprotein layer. So some of the secretions seep into the zona pellucida. Now, when they seep into the zona pellucida, it causes for the hydrolysis of the sperm binding receptors. So we have, remember we stated earlier that the ZP3 has sperm binding receptors to which the sperm binds onto. Okay, now this cortical content, the content of the cortical granules, seeps into the glycoprotein layer of the zona pellucida, and it causes for the hydrolysis of those sperm binding receptors such that no other sperm can bind to the zona pellucida and by extension get into the ovum. It prevents polyspermy. Apart from that, it also causes for the breakdown of the ZP2 glycoprotein, which essentially changes and hardens the form of the zona pellucida such that no other sperm can get into the ovum, okay, preventing polyspermy. So there are two mechanisms by which polyspermy is prevented. First, we have the oocyte membrane block, which is initiated by the binding of the sperm to the sperm binding receptors on the plasma membrane of the oocyte. We have sperm binding receptors on the plasma membrane of the oocyte as well as those on the zona pellucida. But it is when the sperm binds to those receptors on the plasma membrane of the oocyte that oocyte membrane block is initiated, and this basically involves the removal of other sperm binding receptors from the plasma membrane of the oocyte. Okay, now once the cytoplasmic content of the sperm gets into the ovum, it leads to the liberation of internal calcium stores, and this liberation of internal calcium stores does two things majorly. It allows for the continuation of the second meiotic division, which was arrested at metaphase 2, and it also causes for cortical reaction. And we can see how the cortical granules basically prevent polyspermy in the ovum, okay, preventing the entrance of more than one sperm. Now, so far we have considered acrosomal reaction and prevention of polyspermy. It's important for us to take note of the fact that when considering the prevention to polyspermy, is generally classified as the fast block and the slow block. So there's a slow block to polyspermy and there's a fast block to polyspermy. The fast block to polyspermy in humans can be referred to as the oocyte membrane block, which we considered earlier, and the slow block would be the cortical reactions. However, in certain organisms in certain studied organisms like the sea urchin, A fast block to polyspermy is a rapid depolarization of the cell membrane of the ovum from about minus 70 millivolts to about positive 10 millivolts. This rapid depolarization prevents further entrance of sperm cells, and so it prevents polyspermy in that way. However, in humans, this is not exactly the case. The tendency or the probability of polyspermy in humans is further decreased by the fact that the sperm cells that are released from the isthmic region into the ampullar region per time is decreased. Okay, remember when we're considering the hyperactivity in the isthmic region, there's a decreased number of sperm per time that gets from the isthmus, from the isthmic region to the ampullar region. So it's good for us to take note of that. Now, in binding to the ovum, it's important for us to take note of a preferential attachment. So we have a preferential attachment of the sperm cell. It's important for us to take note of the fact that the sperm cells preferentially attach to regions of the ovum that have microvilli. Okay, the sperm cell would preferentially attach to regions of the ovum that have the microvilli. Now remember that when we were considering oogenesis, after the end of the first meiotic division, we have the formation of the first polar body and the secondary oocyte. The major difference between the first polar body and the secondary oocyte is the fact that the secondary oocyte is basically what would potentially form the viable ovum or the viable egg. Okay, and the polar body would degenerate over time, although it also undergoes the second meiotic division, leading totally to the formation of 3 polar bodies. However, during fertilization, we have the presence of both the first polar body and the secondary oocyte. Because the sperm has a preferential attachment to areas of the ovum where there are microvilli, there is a low tendency of the sperm attaching to the first polar body, which is not viable for formation of a zygote. Okay, the first polar body lacks microvilli, so the sperm cell would most likely not attach to the first polar body. Also, in the secondary oocyte, the region where metaphase has been arrested also lacks this. So if this be the region where the metaphase has been arrested, this region also lacks microvilli. Okay, this region also lacks microvilli, and so this means that the sperm cell is less likely to attach around this region. Okay, and this is in order to prevent any disruption to the meiotic division that is taking place. Okay, so the sperm cell would most likely attach at a region probably around here, okay, away from the first polar body and the part where the meiotic division is taking place. Another very important fact that we need to take note of is the degeneration of the paternal mitochondria. Now, the paternal mitochondria is mostly degenerated in the zygote. Okay, now if we take a look at our diagram of the sperm cell, we can see that contained in the middle piece, we actually have a mitochondria. And alongside other cytoplasmic components of the sperm cell, the mitochondria also enters into the cytoplasm of the ovum. However, it doesn't form the functional mitochondrial complements of the zygote. Mitochondrial DNA is from the mother. It's always inherited from the mother and not from the father. And this is because there is a degeneration of the paternal mitochondria, and it's supposed that this degeneration is as a result of the exposure of the inner membrane of the mitochondria. So the mitochondria is made up of an outer membrane, and we have an inner membrane that forms this cristae. Okay, and it's assumed that this degeneration of the paternal mitochondria is as a result of a disruption in the inner membrane leading to an exposure of the mitochondrial DNA to this caspase enzyme that causes for its apoptosis. Okay, so the mitochondrial complements in the zygotes, the functional mitochondrial complements in the zygotes originate from the maternal side, not from the paternal side. Okay, so it's good for us to take note of that. Now, so far we have our sperm having entered into the ovum. Okay, now as I stated earlier, the entrance of the sperm into the ovum causes for the liberation of these calcium ions. And apart from corticular reaction, this calcium ion also causes for the continuation of the meiotic division. Okay, so the meiotic division progresses into the anaphase and the telophase, and then cytokinesis, where the second polar body is formed, and then we have the ovum. We also have the formation of the female pronuclei or the female pronucleus. And also, once the sperm cytoplasmic content gets into the ovum, we also have the formation of the male pronucleus. And so these two pronuclei move closer together. When they come in contact, there is a fusion of the membrane and an intermingling of the paternal chromosome and the maternal chromosome. But of course, before this intermingling happens, it's important for us to take note of the fact that DNA replication actually takes place in each of the pronuclei. Okay, so each of the chromosome is represented as two chromatids. Okay, just as an aside, it's important for us to take note of the fact that a single chromatid is a chromosome, is referred to as a chromosome, and a dual chromatid is also referred to as a chromosome. Okay, so we have DNA replication taking place, and the chromosome is now duplicated, so to say, and is represented as two chromatids. So we have 23 haploid dual chromosomes from the maternal side, and also 23 haploid dual chromosomes from the paternal side, and these two fuse together, forming this conceptus or this zygote that has 46 diploid dual chromosomes. Okay, and this conceptus is now ready for cleavage, okay, which we'll see as we go on in subsequent videos. So far in this video, we have been able to consider the sperm transport, we considered capacitation, fertilization, under which we considered acrosomal reaction, prevention to polyspermy, and fusion of the pronuclei. It's very important for us to take note of the fact that fertilization is very important for various reasons. The gender of the baby, of the zygote, is also determined. A sperm with an X chromosome would form a female baby. Sperm with a Y chromosome would form a male baby. Okay, so there are various importance of fertilization. We have come to the end of this video. If you liked this video, please give it a thumbs up and don't forget to subscribe. Thank you for watching till the end. Bye.